I do want to mention that tonight's lecture is part of, again, our Catalyst of Change series, where we're really trying to talk about some of the big issues that are confronting humanity even as we speak. And so you've heard about the beginnings and life in the outside the universe, outside the solar system, sorry. We've heard a little bit about the biosphere and the challenges that will have. We've heard about AI and the implications that are happening right now as you pick up your phone. And tonight we're going to hear from Howard Yana-Shapiro. So let me just give you a short introduction to Howard. He's been involved with sustainable agricultural and agroforestry systems, pattern recognition, plant breeding, molecular biology, and genetics for over 40 years. He's released hundreds of plant varieties called cultivars into the public domain. He's worked with indigenous communities. He's a former university professor and saw the light, and has also been a Fulbright Scholar and Ford Foundation Fellow. And I could go on, but probably the most interesting thing is that he is working with the African Plant Breeding Academy with UC Davis at the World Agroforestry Center in Nairobi. And he recently also collects and restores classic American, modern Japanese, and Italian motorcycles. So please welcome Howard Yana-Shapiro.
Just before I start, I'd like to make some thanks, certainly to Elliot, the interim dean of the College of Science, to Joaquin Ruiz, the Vice President of Global Environmental Futures at the University of Arizona, to Bo Baylor, who is retiring at the end of this month, who has been a phenomenal inspiration to the way I've worked with the University of Arizona for many, many years. The beauty of what I'll be showing you, I wish I could say I created myself. It's really the genius of Ken Godat and Danny Pagano at Godat Design, who worked with all the speakers to do this. And without their assistance, this presentation could not have happened. The word wicked is a funny word to start a presentation with. But in the world that I live in, the word wicked is not, he's a wicked person. It has to do with some of the greatest, most impactful problems that are incredibly complex that we face as a society, oftentimes impacting human health. So this sets the sort of theory behind what I'd like to talk about today. So it's big systems, it's far-reaching, it's global and it's interdependent. There's no way for a piece to be pulled out and the system will continue. It's nonlinear. It's economic and environmental. And if you look around the room, every person who's sitting next to you, behind you, in front of you, it's shared. This whole notion of what we do is shared as human beings. And 6 of the 17 Sustainable Development Goals deal with things that I find impactful for me on a daily basis. And without trying to solve problems about poverty, about obesity, malnutrition, and food security, about global climate change or human health, access to clean water, and gender equality, which are 6 of the 17 goals, we don't really have a chance to move forward as a society unless we like the fact there will be those that have it and those that don't. And science plays a huge role in making an equanimity of the system. In 2019, and I only refer to papers from 2019 and 2020 because I don't want you to think I'm locked in history no matter how old I look. These are papers that I was very interested in. They deal with algae and algae production. They were in magazines like Plant Science Today, high-tier, high-profile journals. But in 1959, a 12-year-old kid got very interested in this topic as well. And so when I wrote my first scientific paper, you got to start somewhere, right? was in 1959 on marine algae, Food for Tomorrow. And you can read barely some of the commentaries that you'll see. Basically, what I was saying is there another food that we haven't looked at from the ocean? Can we make it cost effective? Can it fill some of the needs? Algae is food. A substitute for space flights. I was already thinking about space flights in 1959. I had a vivid imagination. I also always wanted a watch that I could talk to like Dick Tracy, and now I finally have it. And in my taste area, when we had panels, we thought it tasted like broccoli, but some people felt it tasted like prunes. Quite a different viewpoint. The thing that you must always keep in mind is whether chlorella can be an important contribution to the world food system. And then I was at 50 cents a pound, where products like maize, soybeans, and wheat were at 13 cents a pound. And at age 12, I didn't understand how to make the economics work. So I moved on to other topics and brought me here today. So I'm very interested in humans and nature. And I've spent a lot of years in the field. Some people say I live outside more than I live inside. And it's been in South America originally, but Central America as well, then in Africa and in Asia. And the last 50 plus years, I've spent most of my life in the field. So it's human nature to think about the following things. Do we live with nature like the San people do, hunter-gatherers? Most of us would starve to death, though native people would be able to feed us if they wanted to or felt necessary to do it. We can tame nature, which we have done for more or less 12,000 years. We can conquer nature, which is what we do in great waves, the Amazon on fire, the great tropical rainforest of the Democratic Republic of Congo, the forests of Indonesia. Their destruction is really about conquering it and making it produce something in the wrong place. Or we can exploit it. And I'm part of the exploitation too. I bred plants for years and years. Many of my friends bred plants for many, many years. High inputs, lots of fertilizer. If you apply the fertilizer, 30% blows away, 30% washes away, 30% hits the target, and yet we still apply the same amount the same way, anhydrous ammonia. Or we can change it. And the world I live in today as a scientist is about changing it. We can genetically engineer it, which is complicated in many people's view because of the nature or the story of GMOs. But if we were to take a survey of all the people who take insulin in this room, and most heart medicines, they're all genetically engineered. So somehow it's okay to keep us alive, but it's not okay for us to eat it. And then there's synthetic biology, which I'll talk about later. And then a very interesting topic near and dear to my heart, because I utilize it every day in my labs at UC Davis, is CRISPR, which is gene editing, which has been determined by the United States and Australia and a few other countries not to be in the same category as a GMO. The Europeans had a different view, but that's being reviewed at this point. This is my view. Food production is the biggest threat to the planet. I could tell you it's climate change. That's also true. But if we don't have food, it's going to be a very, very different story. And the global food problem isn't necessarily what you think it is. We have these views of the world. We have only talked about yield for 70 years. How much yield can we get per acre? Can we increase the yield? So your big 10 crops have basically hit their ceiling, their yield potential. So maize is not growing 5 or 10% in production a year unless you add more land, because we've hit this production ceiling. But what if we were to shift that conversation and say, food security, we're going to change the words to mean nutrition security. The fact is, the real problem in the world is nutrition. We probably have enough food if there were not problems that I'll get into later of spoilage or distribution or these sorts of things. But what if you were paid as a farmer on nutrition per acre? And a few years ago, at a meeting at the State Department, I was on the stage with the president of then Monsanto and the president of Pioneer Hybrid, two giants in the seed world. And I said to them, I had this project I was doing, and I would ask them if they would share with me how they worked on nutrition, because I didn't want to know how they worked on yield, just on nutrition. And they sheepishly said, we've never worked on nutrition. So for 70 years of massive efforts, millions and millions of dollars, genius plant scientists, they had never worked on nutrition as the foremost thing. And just a few things. This is global food production by the numbers. 70% of biodiversity loss in the world, and some people say it's only 65, and some people say it's 75, but let's just use a rounding number like 70, of the biodiversity loss in the world is caused by modern agriculture. 70% of the fresh water use is used in agriculture. And the water that we're using is actually Cleopatra's bathwater, because water is a closed system. It doesn't go away. It evaporates, it goes up, it moves around the world, it falls, there's rain in a different place. 25% of the global gas emissions are from agriculture, 25%. 85% of the marine stocks of the world are fully exploited at this point. It uses the most chemicals in the world, and we lose 50% of the topsoil loss in the world has been caused by modern agriculture. These are not statistics that make you feel good at night. And we have no idea how to feed the planet without frying it. This was an editorial in the Washington Post in August of last year. Humans have to get much better at growing more on less land. And this has become a clarion call from everyone in what I would call it resilient agriculture. Resilience, by definition, is the ability to respond quickly to a problem. The definition for sustainability was written over 35 years ago. So I want the ability to respond quickly. I don't want a definition that's 35 years old, just like you don't go to a dentist who uses the same tools from 35 years ago in the techniques. We need solutions. Jason Clay, who is a well-known author in the area of sustainability and resilience from the World Wildlife Federation, has said no single actor or publication will make the global food system more sustainable. And wisely, he points out that there's 7.5 billion food experts in the world. I look at this room, every one of you is a food expert. So how are we going to coalesce around common goals? How will we not have this be a wicked problem? The speed of change is typically faster than our response, and it has been for decades. It takes 30 years to bring a new tree onto the marketplace, or 25 years. It takes 7 to 10 years to bring a new annual food crop to the marketplace. We need to fix that. And there are 3 ways we can look at it, and I'll mention a 4th way at the very end. Through computing, through analysis, and through genetics. And by trade, I refer to myself as a gene jockey. I work with genetics. That's what we do. So a chip that rivals the human brain, and I want to make clear here, I'm not talking about a chip that acts like a human brain, but has the capacity of 1.2 trillion transistors on a chip. 1.2 trillion transistors on a chip. And I know last week you had a presentation on AI and machine learning, and I don't want to contradict what he was saying, because this is not about outdoing the brain. The brain is a unique instrument by itself. But if you look around 1990, there were 10,000 transistors on a chip. And around the year 2000, it was around 50 million. And today, it's 1.2 trillion. Now, if you look in your phone and you take it apart, which you really shouldn't do unless you know how to put it back together, and you see a chip, and start to imagine what 1.2 trillion transistors means. It's almost unfathomable that we have the technology to actually do that. Analysis. There's a piece of kit that we use a lot called a mass spectrometer, or mass spec. In year 2004, per dollar, you could get about 3 passes on a mass spec. By 2009, it was pretty much the same, but you can see that by 2019, which is not on this chart, we're over 1,000 views per $1. Again, there's no reason why this can't open the doors and solve the problems. And because I do a lot of genetics, this is very important to me. The cost today in 2019, we just did the calculations last night, it's 10 to the minus 9 compared to it was in 1980, to give you an idea of the scale of what we're discovering. Now, no one in the dendrology department can answer this question. How many of you can identify this seed? Shout out. Rice, no, come on now. Imagination. All right. It weighs 1/600th of a gram, to give you an idea. 2 days old. 2 months old. And now the surprise, 1/600th of a gram grew General Sherman. It weighs as much as 6 747 airplanes fully loaded with passengers, fuel, and baggage. This is megabiota. I know you heard that term a couple lectures ago, and the reason I say it, this tree is 392 feet tall. There are actually taller trees in the area around this particular redwood. Imagine 392 feet tall was grown from 1/600th of a gram. Think about the information that was packed in to the germ or the embryo of that seed. And at 100 feet up, there's things that happen on the surface of that tree that are different at the ground. And at 200 feet up, it's different. And 300 feet up, it's different. And because they now have drones and people climb up these with permission of the federal government to do research, at the top of some of these crowns, there's bowls of dirt 5 feet across with amphibians and other things living in it. That's megabiota. We have never even understood how that could happen. So I love the fact that that little seed produced this. That's why for so many years I've been involved with seed production. And this is a maize seed, which every one of you knows. But when you look at it, the video didn't work. Let's hope it gets better. It was going to spin around and show you the germ. Now, where does maize come from? It didn't look like modern maize when it started. Nothing looks the way we have it now. If you study the image of teosinte, this is teosinte mexicana. There's a number of varieties of it. Some witty person decided, I'll start making selections. And they came to what was known as a teosinte corn hybrid. There was no corn. Corn didn't exist 7 to 10,000 years ago. It all came from the Oaxaca Valley of Mexico. So some witty woman or man started making selections, saving seeds in different piles, saying, I'll grow this, you grow that. And eventually, it became modern maize. And that's what a seed of teosinte looks like. But inside that seed is the germ or embryo which carries information forward, generation after generation after generation. What we do as servants to the plant is make selections. I like this one better than that one. I'll grow this one. Somebody else will grow that one. And in 1931, or maybe it was 1937, excuse me, during the Farm Security Administration, they sent a number of photographers around the world, Margaret Burke White, Ben Shawn, Dorothea Lange, Arthur Rothstein. These are famous photographers and painters to record America. And this was the state fair. And this man had selected this maize. If you look at it, and I know the picture's a little fuzzy, if you look at it, every ear seems to be an exact copy of the other ear. That's what hybridization does. You make a choice. Now, you can also have a problem that when you select too narrowly, then your particular plant is susceptible to disease at a very high level. So when I grew up as a plant breeder, this is what I could see. We would trudge through the fields with notebooks and pencils, and we would take notes that this plant looks perfect. You'd put a tag on it, so you would come back and harvest it. It was all observational. But this is what I can see now. 2 times 10 to the minus 2, the ear of corn, the kernel of corn. The reason I'm showing this is how deep we can go in seeing things that we couldn't see very long ago. Condensed chromosomes, that's what a real chromosome looks like. And this is the scaffold that holds it together. And this is a histone. And now DNA. Never in my life as a young scientist did I ever think I would see any of this. And it's totally commonplace now. Freshmen in college, seniors in high school see this. But it wouldn't have been possible without a series of heroines and heroes who worked constantly against all odds to create what made a toolkit for me to be a guardian of plants. And I'll start with a historian who makes a very interesting point. We did not domesticate wheat. Wheat domesticated us. And the reason I think it's important, because the plants were always there, we were the ones who were evolving around the plants. We made some selections. But because of these selections, we were allowed to domesticate and have a society. And it began a long time ago. You have the cognitive revolution, more or less 70,000 years ago, when we learned to really how to communicate and think and understand and make decisions. There's the agricultural revolution, which was 10 to 12,000 years ago, depending upon who you think has done the best work in dating plants. And then there's the scientific revolution, which is only 500 years old. It's like a baby. But with the advent of things like AI and machine learning, there's another revolution coming that I don't think we fully understand just yet. Barbara McClintock, one of the greatest scientists of all times, she looked at Native American corn and wondered why they had different colors. Because the modern corn was yellow or white or dark yellow. And she started to think, I wonder if it's possible that those genes could jump around. The pollen that fertilizes from the female plant to the male, from the male plant to the female plant, would that carry information? Would that carry knowledge? Would that carry a future of that particular plant? And that became known as transposons, or jumping genes. When you're in high school, your science teacher calls it jumping genes because you probably won't remember transposons. And Barbara won the Nobel Prize for her effort. Frederick Sanger, one of the few individuals ever to win 2 Nobel Prizes. Not that having just one wouldn't be exciting. He was a biochemist. And what he did was he discovered that he was able to read the molecules of life. And then he figured out, but if I had a machine that could do that, let's call it a sequencer, and I'll call the first one the Sanger sequencer, then we could really invent something. We could really go deep into our knowledge. And then along came this little funny company called BGI. The average age of the employee there is 27 years old. There's about 5,000 of them in Shenzhen, China. This is one of their headquarters. It goes all the way into the mountain. It's the Chinese germplasm collection. In 2018, they came out with a machine called the T7 DNA sequencer. It can do 60 human genomes a day. And what is the comparison? To the human genome, which took 13 years and $1 billion to complete. Imagine what that means for understanding information. You think that's impressive? 5 days ago, this happened. They came out with a new machine that says they will give you a human genome for $100. This is the Holy Grail. Because for $100, if you went into a hospital to try to determine what was wrong with you, normally they wouldn't sequence your genome. But now for $100, it's just part of the process. That means we'll know more about someone who's ill very quickly in life. And this machine can do 100,000 human genomes a year. So imagine if you had 20 of these in a room, how much information you could generate. And I have a thing to show you. I usually carry one of these in my backpack everywhere I go. It's the same as in the picture which was taken yesterday, right out here. This is a sequencer. This is a sequencer made by an upstart company called Oxford Nanopore. This is called a MinION. I can go into the field, carry a kit with me in my backpack that will help me extract the DNA, and with a simple pipette, I can fill this up, close the top, take this cord, plug it into my computer and my USB port, and I can sequence something. Did I ever think this was going to happen? M.S. Swaminathan, who partnered with a guy named Norman Borlaug, they were asked a very simple thing. Wheat grew tall. You know, the first wheats, the Eimer wheats grew 5 or 6 feet tall. The wind would blow, the rain would come, they would fall over. It's called lodging. And there was problems with wheat lodging in India and Pakistan. So the two governments asked Swaminathan and Borlaug, fix the problem. What did they do? They developed a dwarf wheat that wouldn't fall over in the wind. It was strong. It had really good roots. The rain wouldn't make it fall. And by doing that, Swaminathan won the World Food Prize. Borlaug won the Nobel Prize. He was an agronomist. There's no Nobel Prize for agronomy, so he got it for peace, because what happened, he created the Green Revolution, which drove the very large chance of a massive starvation due to not having enough wheat for Pakistan and India to making them nearly self-sufficient. Now, the Green Revolution has its critics. I'm one of them. And I know Borlaug when he was alive, and I talked to Swaminathan periodically, and I collaborated with Borlaug on a long-term project. Borlaug never thought what he designed and the plant he grew and the methodology for the system would last from the late 60s through 2020. It was a solution to a problem. But what happens to us sometimes? We get lazy. And nobody improved it. And so Swaminathan said, what I really wanted to do was to create an evergreen revolution, not just a green revolution. And now people are working on improving it. Gert of Kush, his office is next to mine at UC Davis. And Gurdjieff won the Wolf Prize and the World Food Prize. As he said, agriculture scientists and geneticists need to work overtime to meet ever-growing human needs for food and to alleviate hunger and poverty. This is IR 6.4. This rice is eaten by more people than any other crop in the world. Imagine what it took. You're starting at the extremities of this diagram, making all these crosses, back crosses, more crosses, more crosses, more crosses. The dedication in the mind's eye to actually go after what was necessary to produce rice that was more robust, higher yielding than anything that ever existed before. Again, to forestall starvation. And that is IR 6.4. Frances Arnold, Caltech, chemist. What did she say? In the lab, we're discovering that nature can do chemistry that we never dreamed possible. Imagine the Nobel Prize winner saying that nature is smarter than we are. And so what did she do? She created enzymes that have replaced toxic chemicals in many industrial processes by making them synthetically, understanding what was going on. She bioengineered enzymes to do stuff that had never been done before, to replace toxic chemicals, to help improve the quality of life for human beings. And down the road at another University of California outlet, it's called University of California, Berkeley. UC Davis was the Berkeley farm till they determined that farming really wasn't a good science, so they let it go, and we became the University of California, Davis. And the last few years, we've had more federal dollars, international grants come in than Berkeley. We've surpassed them in research. But Jennifer Doudna, with a partner named Charpentier, developed a little funny thing called CRISPR. The power to control our species' genetic future is awesome and terrifying. It's awesome and it's terrifying. Deciding how to handle it may be the biggest challenge we've ever faced. So it's not just scientists now, it's ethicists, it's social interactions with human beings. This is not a simple thing. So I'm going to go through a few slides that describe CRISPR. It may be more than you want to see, but I think it's important because there's so many misgivings or misunderstandings about what CRISPR is. Before CRISPRs, there was something called zinc fingers. And zinc fingers was a very simple process, protein-based targeting. It was a mature technology, largely worked. Then we have talons, which came after zinc fingers, which actually did more modular work on understanding how they could protein base. And then came CRISPR, CRISPR-Cas9. There's Cas3 and Cas12a. There's a whole variety, and people have CPSIs now, which are different than Cas9. But what did it do? You have a target DNA, you have something that guides it there called guide RNA, and then you have this enzyme, Cas9, which cuts it and repairs it. So here's a little bit more of an explanation, because I want you to feel comfortable when someone asks, are you afraid of CRISPR or are you thinking it has potential? So here you see your guide sequence, you see your guide RNA, you see your cutting site, the little mouth that's cutting. Then you see your target DNA, your Cas9, and then you see a repair. One more. It can also work on viruses, not just in the sense we're using it right now, on the bacterial immune system. And there it is. You cut it. It's truly like a scissors. You just cut the DNA, splice it together, and repair it, and you move on. You haven't added anything to it. You've only taken something away. Now, why was I interested in it originally? For many years, I had the distinction and pleasure of being the chief agricultural officer for a company called Mars Incorporated, the giant confectionery company and the world's largest pet health and nutrition company. And we have a tree called the Theobroma cacao tree. And that tree suffers mightily from every disease because it is not highly developed. And there's a particular problem in West Africa called swollen shoot virus, which has 16 different variants. And it could wipe out the crop. But using a technology like CRISPR, we might be able to go into the plant, cut out the receptors to the CSSVD, put it back together so the tree is resilient against this particular virus. That's how I got interested in CRISPR. That's how I got to know and work with Jennifer Doudna. And this is how it works kind of in real time, if you will. There's the scissors, and it works in many, many different crops and animals and plants, 60 eukaryotic species currently, but soon it'll work in everything. And now imagine in 2011, there was about 70 papers written on CRISPR. And in 2018, the last time a tally was done, there were 17,000 peer-reviewed scientific papers on CRISPR. One other key factor, it's democratized. At the University of Arizona, there must be a dozen labs working on CRISPR. UC Davis probably has 50 labs because of the scale of our veterinary school and of the ag college. In Europe, which has banned it, every country has hundreds of labs working on this because it has a promise. But as Jennifer Doudna said, it's terrifying, the promise, especially when we start talking about human beings. And the area where it's being trialed at this point is on sickle cell anemia, which is a terrible, terrible disease that hits certain parts of our population. So just 5-6 months ago, a guy named Ron Platt published a paper that just making one move on a gene wasn't enough. He can make 25 moves in a single gene. This is repair beyond our imagination. He thinks he can do hundreds. But then there's always the question, if a consumer does not believe that key information about a food is being hidden from them, they may question the food's safety, quality, or supply chain. So the public has to be brought along. You all have to become informed, whether you agree with it or don't agree with it, CRISPR and its applications. You must be informed so we can have a dialogue that is not based on mythology but on fact. And one of the other things that has struck me is the issue of chronic hunger and malnutrition. And most of us hear about it, and most of us talk about it, and then we go about our daily lives. But a few years ago, a young professor, an assistant professor at UC Davis named Christine Stewart, made a little 5-minute flash presentation. At UC Davis, I sit on the Dean's Council. And when she finished, I followed her out into the hallway and asked her a question. Is everything you said true? Can you show me the data? It's hard to believe what you've just told me. And I'll explain that to you in a moment. She goes, yes, it's all true. I said, I'm coming to your office tomorrow morning at 9 o'clock. I want to see all the data. And what she told me about was this condition called stunting, which is caused by chronic hunger and malnutrition. So most of us know stunting as a person is small for their age and their weight is low. What you don't know is their neurological development will never, ever, ever be like anyone in this room. They won't be Barbara McClintock, Frances Arnold, Jennifer Doudna, M.S. Swaminathan, Norman Borlaug, Gurdjieff Kush. They'll never be like any of you and me. And then economically, they're disadvantaged and they will die early because their immune system is not robust. I was flabbergasted. I remember coming home to my wife Nancy and saying, I've never been so depressed in my life that I've actually now learned that there's a condition that is so pervasive, and who's doing anything about it? So, like a curious person, I started looking into it after meeting with Christine Stewart. You can see by the chart, lots of red means 40 or more % of the population under 5 is stunted. Shocking numbers. America is less than 5%, but since this was done, we think it's around 7% of the population. Africa, it's 37%. India, it's 48%. You know, I'm a world citizen. I'm saying to myself, how can I possibly not try to solve this problem? But I've worked the most in Africa, so I'll try to solve it in Africa. And if it works, it'll be mimicked to other places in the world. But I don't know how to do it. I got no place to do it. I've got no money to do it. But I had this idea. If we know that food insecurity is worse in countries where there has been conflict, that's another part. And if you look at this little diagram of food prices and food riots, and you look at that little area around 2010 to 2012, Tunisia, Libya, Egypt, Syria. It's the Arab Spring. It was caused as much by food riots because food was not available. And in Egypt, what happened? They grow a lot of wheat, but not enough to supply the community of bakers. They get it from the Ukraine. What happened to the Ukraine? They had a crop failure. So something as simple as a crop failure in the Ukraine flutters its wings and causes a food riot in Egypt and in Syria and in Tunisia, and over 12,000 people died, but you know what happened after that. I'm thinking about all this stuff. Okay. 1 in every 5 Africans is hungry chronically. 59 million people under the age of 5 are stunted. 13.8 more are affected by wasting, which is a version of stunting. 9.7 million under the age of 5 are overweight, mostly in the north in the Arab states. And 38% of the women of reproductive age are affected by anemia. How do you fix this? You fix it by fixing the food system, right? You fix it by fixing what they eat. Since you can't fix the politics, you fix the food they eat. You improve the basic structure of the food system in Africa. So we did a survey, sent it out to 100 people in Africa, food scientists, women's groups, women activist groups, nutritionists, wide range. We sent 100 surveys out and got 106 surveys back. And those of you who give surveys know if you get 23% back, you jump for joy. The problem was now we had so much data, I had to hire someone to analyze the data, and we put a hierarchy together. And we decided that we would take on 16 crops. Remember, in my era, if you did one genome in your lifetime, hang your hat on it. So I did the cacao genome, sponsored by Mars Incorporated, USDA, and IBM. And then we had a problem with peanuts, so I initiated the peanut genome. And I thought in my lifetime maybe I could do 6 of the 16. Other people would follow through. So then I went to this little funny company called BGI in Shenzhen, China, and I told them I needed their help to sequence 16 crops. It was on Thanksgiving Day in 2014. And the director, Jian Jiang, said to me, well, 16 won't fix anything. I'm going, I came to do 16. I can't do more. He goes, 32. I say, okay, 32 is fine. I can do 32. I had no idea how to do it. Then he said, but in thinking about it, I don't think that's enough. We ended up with 96 crops. Because they were going to do the heavy lifting on the sequencing, I would build a laboratory in Nairobi, which I had no idea what I was going to do, but we would do all the resequencing, which is looking at many varieties of the crop. So then I went to the African Union, and they said, okay, nice number, let's make it 100. 100 is better than 96. And I said, okay, I'll do 100. And then a person, the president of Ghana, got up and said, I think it was Ghana, you forgot one crop. Now, 100 was pretty well considered and well conceived. This were the main food crops of Africa. And I sheepishly said, which one was it? He said, the baobab tree, because it's the icon of Africa. Just to give you an idea, the baobab genome is about 6 to 8 times bigger than the human genome, and it's never been looked at. It's almost impossible. So I hope before I die, I'll finish the baobab genome. So let's say you want to improve a carrot. Why would you improve the carrot if you were talking about nutrition? Well, here's why. Vitamin A. The reason you all can see pretty well, and I can see pretty far back, is that I have normal vision at night. On the other side of the screen, that's what night blindness looks like at night if you have vitamin A deficiency, which is rampant in Africa. Leafy greens are important for your red blood cells. That's what your hemoglobin actually looks like. That's what heme looks like, and that's what iron looks like. Healthy blood cells. That's anemia from a lack of iron. So let's improve all the leafy greens in Africa and boost the iron content. Seaweed. Many people on the coastal parts of Africa eat seaweed. And why is it important? It's important for a very simple reason. Iodine. Stops cretinism, which is irreversible as well as stunting. And one more, which is really critical, is zinc. Because zinc, if you don't have it, you have stunted growth automatically. So why is it important to do it for the rural foods? And when do you have to have a young woman eating these improved crops? Not when she becomes pregnant, but maybe 750 days before she's pregnant. They have to become part of her diet. They have to become part of her life. So that when she becomes pregnant, she's not looking for folate, she's not looking for iron, she's not looking for zinc, she's not looking for iodine. She's robust in her diet. 101 food crops. So what do you need to do? Well, we need to breed crops that are for nutrition. But if they aren't high yielding, nobody will grow them. What else? They have to be resistant to pest and disease, water efficient, and nutrient use efficient. And if you think about the most important one, maybe resilience to climate change. This is what you have to do in a crop. Now remember, most crops in modern breeding aren't done this way. So how are you going to do it? Well, you start the African Orphan Crops Consortium, and you start the African Plant Breeding Academy to do all of this work. But you don't want to have the students come to the United States, to UC Davis, and decide to stay. You want to have them trained in Africa. You want to pay for every single cost of the training, so there's no excuse for the poorest university not to send their smartest people to this. You want to take it and make it available, and you want to make it competitive. So we started the African Orphan Crops. And a woman who was at this time, Rita Larte, was at the University of Ghana at Legon, said, if you do anything for me, make sure you have as many women as possible in your class. So we decided we would only have classes with 50-50. 50% women and 50% men. And it was hard to convince people that if you were admitted to this academy, which meets 3 times a year for 2 weeks, taught by some of the best people in the world that we bring in, and everything is paid for, including if you don't have a computer that's good enough to do the calculations in R, because we use R for all of our statistics, because it's open source, then I'll give you a brand new computer. So my hero is Ibrahim Mayake, who is the CEO of NEPAD, which is the development arm of the African Union. I went to Ibrahim, and this is what he said. Malnutrition can have a devastating effect on population. If you can fix this, Howard, I'm behind you. I'll be your sponsor at the African Union at the President's Council. And then my theory was, this is a huge leap towards diversity if we can do this. This is a new day. And my colleague Tony Simons, who's the DG, Director General of C4ICRAF in Nairobi, said, I'll give you a space. I'll give you all the space you need to teach the class and to house the molecular biology laboratory. For the continent that is the most malnourished, the poorest, the most rural, and the least forested, this project gives a future. And this is graduation of class 3. And in the back row, there's a head sticking up. That's Bruce Walsh. Right over there. Bruce Walsh is one of the greatest plant scientists ever lived, and he teaches at the University of Arizona. And he couldn't be here tonight, unfortunately. The way he cares for these students, and the way Rita Mum, who runs it, and all the other teachers, the way they bring the best out of these best in Africa cohorts in this class is astonishing. A few statistics. It's an uncommon collaboration because I don't know how to go straight. I like to go nonlinear. We opened it at the World Agroforestry Center. We've graduated 115 mid-career scientists. They have raised by themselves over 18 million euros in European competitive grants post-graduation. We've established the network of African plant breeding. We have 238 simultaneous breeding programs going on, Pan-Africa in 25 countries. We've established the best lab in Africa. We're resequencing 100 times every one of the 101 food crops to have a database of 10,100 genomes. And we've established a small grant fund to give the graduates money to go back to their institutions and do this work. And this is what it takes to do it. This is my crazy quilt. Of course, Mars, I'm very proud of them being the first participant, NEPAD, World Agroforestry, and then 3 of the largest equipment suppliers. Because when I started, Thermo Fisher was the number 1 in the world for sequencing. And in 2 years, that became archaic. So I went to Illumina, and they gave me a HiSeq 4000. And in 18 months, that was archaic. These are $1 million machines. So now, I'm working with Oxford Nanopore. And I'm using the big brother of these machines, called a MinION, which is 24 of these booted together. And we're doing resequencing at a cost that makes it possible to get this job done. A few more things in closing. As soon as I started this project, someone came to me and said, well, that's great, but you didn't solve the aflatoxin puzzle. I said, please, really? Can't you find someone else to do that? Aflatoxins are naturally occurring mycotoxins. In a good year in the United States, it's only a $40 million maize loss. In a bad year, when the plants are infected, it's $1.4 billion in losses. And we have the best technology and the best storage and the best seed and the best everything. Imagine Africa. It's found in cassava and cacao, maize, peanuts, almost all spices. This is what it looks like. 4.5 billion people ingest it on a regular basis. That's more than all other communicative diseases put together. It causes liver cancer. It's horrific. 700 people died a couple years ago from eating mycotoxin maize. I travel in Africa, I buy peanuts. I wonder how much aflatoxin is in this bag of peanuts. Or if you roast some corn on a roadside stand in Africa, how much mycotoxins are in this peanut? Global consequences. Who's affected? 4.5 billion people regularly, but on a daily basis, 1.5 billion people. It affects everything. Economics. It affects the global access to food, nutrition, human health, premature death of women. The highest level of liver cancer, maybe in the world, caused by this. And it's small. Small amount will impact you terribly. 20 parts per billion is the FDA limit. It's 10 parts per billion in Europe. So 1 part per billion equals, imagination-wise, 1 second in 32 years. That moment in 32 years. So Usain Bolt runs the 200 meters in 20 seconds or so. He runs it 1 time in 32 years. That's a toxic dose of aflatoxin, and people are eating it every day. So I said, okay, I got to fix this. Remember, I don't know that you can't do stuff. I talked about computing, I talked about analysis, and I talked about genetics earlier. So I'll reframe something that was talked about last week. Artificial intelligence and machine learning. And why? Because I sat down and thought, I want to take aflatoxin B1, I want to neutralize it, and have a safe food after that. Because if you get clean food out of the field and you put it into inappropriate storage, aflatoxin will invade it. It's like putting a sign up, aflatoxin, come here. All right, how do you do it since this doesn't exist in science? In 2017, this didn't exist in science. So I went to the FAO and the World Food Program, and the UN Sustainable Development Goal is end hunger. And then in the fall of 2017 was World Food Day. So what we launched on World Food Day is the Folded Aflatoxin Puzzle online. It's a game you play. It came out of the University of Washington in Seattle. It's backed by the computers at Northeastern University in Boston. And this puzzle is based at the Genome Center at the University of California, Davis, in synthetic biology. So what do? We put out a call for all the gamers in the world to play this game. And why gamers versus scientists? They don't know you can't solve the problem. They don't know that you can't take proteins and twist them around and create new enzymes to detoxify aflatoxin. So this is what the game looks like if you were to play. And I encourage you all to go and sign up. These parts are all moving around, and someone with their cursor is taking a piece out of, let's say, this region and moving it over there, and then taking another piece and adding it to it. So what happened? since fall 2017. 454,000 participants have played the game, more than once. They've turned in 1,800,000 submissions of proteins that they have manipulated to create new enzymes to detoxify aflatoxin. 100 look pretty good. Not bad, you know. We sent them to a company called Thermo Fisher in Germany. They looked at it and said, okay, 10 are now have been moved into testing. So in the last 70 years of industrial agriculture, no one has made a move on this. And in 2 years, more or less, we have 10 synthetic biology moves that we're trying to make. Here's the other secret. It's in the public domain. Nobody owns it. Anybody can produce these in a beaker in the most fundamental lab, dry it, and then put it on crops in storage. And we believe that in 18 months, we'll have more than one solution to detoxifying aflatoxin. Thank you. It's also fair, I should say, that the first crops from the African Plant Breeding Academy will be hitting the market early this year. And it's the work of a brilliant woman who's in Zimbabwe working with essentially very little support because it's a very poor country. But bambara beans, which are the largest source of protein for that part of the world, she's improved already in 4 years using her knowledge. The speed of change is faster than our response, but I want you to know we are rising to meet the challenge. The one thing we must do, and this is an Oromo proverb from Africa, you can't wake a person who's pretending to sleep. Thank you.